Hello and welcome to your final lesson of year 2. So today I'm going to talk to you about immobilised enzymes. Lesson objectives then we'll look at how immobilised enzymes work and explain some of the processes for which those immobilised enzymes can be used for. So just to give you a bit of an overview of what an immobilised enzyme is. So what do we mean by that? It's an enzyme that is held separate from the reaction mixture. So the reaction mixture is the substrate molecules that you're going to add to that. So the substrate molecules can actually bind to the enzyme molecules and the product will be formed and that will go back into the reaction mixture, but the enzyme is going to remain in place, so it doesn't actually become a part of that reaction mixture. It stays immobilised, fixed in place. So we're going to look at various different ways in which we can immobilise enzymes. We've got adsorption that we'll have a look at. We'll look at cross-linking, entrapment and covalent bonding as some of those. And we'll look at some of the processes that those enzymes are used for as well. So there are a number of reasons why people use enzymes in industry. First of all, because of their specificity. So they've got a very specifically shaped active site, complementary to a specific substrate, which means then they can create those products. And actually, some of the alternatives to enzymes can be extremely expensive. So they might require metal catalysts, very high temperatures, very high pressures, and so actually using enzymes is much better. But they do need to think about this process that's called downstreaming, because obviously what they don't want is for those enzymes to contaminate the product that's being produced. So downstreaming then is the process of removing and purifying the product. So they separate it out from the reaction mixture. And if you're using immobilised enzymes, then because those enzymes are fixed in place, this downstreaming process is much cheaper. They don't have to try and filter out the enzymes from that product because the enzymes have been fixed in place. So there are certain things they can do to try and check for contaminants, and we'll think about that a little bit later. But yeah, you get a lot less contaminants with immobilised enzymes. So we've looked at I mentioned one of the advantages already, which is that you've got these lower downstreaming costs because those enzymes are fixed in place. The enzymes then immediately available then for reuse as well. And the enzymes are actually a lot more stable, so it means that they can do those processes at slightly higher temperatures because when they're immobilised in place, it protects the active site and it'll stop that active site from changing shape. So you can get away with it being slightly higher temperatures, and if you get slight changes in pH, so sometimes with the products that are formed you get pH changes, but a slight change in the pH, it won't actually affect the tertiary structure of that enzyme, so it will continue to function. There are disadvantages though. It's more expensive to set up initially, less active because the enzyme's not moving around. So obviously for any kind of enzyme reaction, you rely on these successful collisions between the enzyme and the substrate molecule. But if your enzyme is fixed in place, then it's just the substrate molecule that's more moving around that can then go into the active site of the enzyme. And if you do get contamination, it can be pretty costly. So you might need to discard the entire batch if there's any kind of contamination. So one of the ways in which enzymes can be immobilised is through a process of adsorption. So with adsorption, you've got porous supports such as clay particles, and you'll have these non-covalent bonds or ionic bonds then between the enzyme here and that porous clay particle. With covalent bonding, Then very similar, so again you've got porous substances, things like clay particles, but here we've got covalent bonds that are holding the enzymes in place. So this is actually much better. You've got a lot lower risk of contamination with covalent bonding because they're held in place by those covalent bonds. There is entrapment, so that's another technique that can be used where it gets trapped in a network of cellulose fibres. Again, your substrate can pass through into the cellulose fibres to get to the enzyme molecules, but because they need to travel through, because they need to diffuse through the medium that it's trapped in, this can take a little bit longer, so it's not as efficient as some of the other ways of immobilizing enzymes. And then there's membrane separation. So membrane separation is when you have a membrane then in between your reaction mixture here with the substrate molecules and the enzymes are on the other side there. So the substrate molecules are small enough to be able to diffuse through this membrane, this partially permeable membrane, and the product can get back as well. But the enzymes are too big to actually diffuse through, so they remain on this side of your membrane. So we're going to look now at some of the uses of immobilized enzymes. So one of the really important uses is the production of new antibiotics. So they'll get known antibiotics, things like penicillin, and they'll feed that in, and then you've got immobilized enzymes, this penicillin acyclase here, held in these alginate beads, these like jelly beads that are in this reaction vessel. And as they go through, the penicillin then, which is the substrate, will go into the active site of the enzymes, and it will convert the penicillin into a number of different antibiotics. So amoxicillin is produced in this way, ampicillin as well is produced in that way, and there are other variations as well that are also produced in using this method. So glucose isomerase then is when they either convert glucose to fructose or fructose to glucose. And that's used a lot in the food industry. So things like jams, so production of jams, things like tomato ketchup, all of these products actually contain fructose that's actually made through this process where they've converted the glucose to fructose using the glucose isomerase. Another example from the food industry then is the use of lactase. So they can use these immobilized enzymes then to convert the lactose to glucose and galactose. So here we've got our lactose molecule here, and then we can hydrolyze that, so the lactase will catalyze the hydrolysis of that lactose to give rise to glucose and galactose. So we were going to actually have a go at doing this, and we were going to actually test our product to see how pure it is. So obviously we could test for glucose, that's the product that we're looking for, but we can also check for contaminants. So contaminants which potentially could be protein. So we could test for proteins as well with those, with the products that's formed. So we were going to use glucose test sticks to test for the glucose, and for protein we can use bio reagent. And this is the alginate beads that we would have made if we'd have been able to have a go at that practical. So another example of an immobilized enzyme that's used frequently then is the aminocyclase enzyme. So that's used to produce these L amino acids, that they remove the acyl groups. That's this group here. They remove that acyl group from the N-acyl amino acids to create these pure L amino acids, and that's used for lots and lots of different things. So used in cosmetic industry, used in agrochemical compounds, animal feed, human food, and pharmaceutical industry as well. So lots and lots of uses for the aminocyclase. We've got nitrile hydrase, so that converts nitriles to amides, and that includes acrylamide. So you might remember when we were doing the manipulating genomes topic and we looked at electrophoresis, that I said about how proteins were separated. So they're separated using a process that's called PAGE, which is polyacrylamide gel electrophoresis. So that's how that acrylamide that's used in PAGE is actually formed. So it gets converted to this polyacrylamide to make those polyacrylamide gels. And this can also be used in water treatment plants. So you can see here that we've got lots of organic matter in this liquid before, and then if we add the acrylamide to this mixture here, then afterwards you can see that when it creates that polyacrylamide and it clumps together, it actually also traps the organic matter. So this is sort of one of the steps that can be used sometimes in purification process for water purification. Glucoamylase, then another example, then converts short chains of glucose, so short chains of glucose molecules, like this one here, into glucose, so it drops it into, or hydrolyzes it. So that can be used for fermentation process, used to convert starch pulp to alcohol, and the alcohol can be used to produce gasohol. So that's a biofuel that you could have. Also used to make starch for, convert starch to high fructose cornstarch. So we can compare immobilized enzymes and free enzymes. Free enzymes, then, the active site is always accessible, whereas with immobilized enzyme, the access to the active site is a little bit slower because they have to diffuse through the medium if it's entrapment, and that can take time. But also the immobilized enzyme is fixed in place, so that takes time as well for the substrate molecule to actually bind to it. So that means that with the free enzymes, you'll have much more frequent collisions because the enzyme and the substrate molecule are moving around, so you're much more likely to collide with one another. Whereas with the immobilized enzyme, you've got less frequent collisions because it's just the substrate molecule that's moving around. More susceptible then to changes in temperature with a free enzyme. So that's one of the advantages then of the immobilized enzyme, because they're less susceptible to change, because that immobilization process then provides protection. Free enzymes, though, you have got much higher downstreaming costs because they need to be able to remove the enzyme from the product, whereas with the immobilized enzymes, the enzymes are fixed in place, so you don't have to pay as much for purification of that product. Free enzymes, it is easier, it is cheaper to set up, it's much quicker to set up as well, because you just put the enzymes with the substrate in the reaction mixture. Immobilized enzyme, it is more expensive to set it up initially to get that bioreactor started. So you've got a couple of questions in your booklet that you can have a look at. One of them is to do with papain. So it's a protein hydrolysing enzyme found in pawpaw fruit and pineapples. And if you look at the graph here, you can see how the free enzyme and the immobilized enzyme behave at different temperatures. So with the free enzyme, you can see that you've got your optimum there at around about 65 degrees. With the immobilized enzyme, then the optimum is at 80 degrees. And with the free enzyme, you can see that it starts to denature, so the enzyme activity decreases, whereas with the immobilized enzyme, yes, it does start to decrease, but it's not decreasing as rapidly at that point there. So we've actually got lots and lots of links with brewing, because we thought about brewing as part of this biotech topic. Obviously, there's links with the use of enzymes. We've got links with the plant responses topic, biological molecules topic, respiration topic, fermentation topic, and the idea of selective breeding as well. So lots and lots of links with the brewing industry. Good. Well, I hope you enjoyed your final lesson. I just thought I would make a quick video for you for that one, and